All right. It's nice to see a great number of people joining this session today. Hello and welcome to the third session of the CELEC Starter Culture Lecture Series. My name is Fabian. I'm a co-founder and also board member of CELEC Germany. And as you have realized, today's session, and actually also the next two sessions, will be held in English. Then for those who did not join the last sessions, this CASC lecture series organized and hosted by the nonprofit organization CELEC Germany. And we ourselves, as a platform for CELEC in German-speaking countries, aim to inform and also aim to connect the industry and people in the industry for the further development and acceleration. And in today's session, we will do a little deep dive into the technical aspects of precision fermentation, representing kind of the second pillar of cellular agriculture. So basically next to cultivated meat, we had this session last week. And for this purpose, I'm happy to introduce you to Professor Marius Henkel, who became the worldwide first professor for cellular agriculture, specifically at the Technical University of Munich. And also next to Marius, Sybe Hartmans, who is a fermentation expert at the precision fermentation company Cowboys from Belgium. Marius will start today and then hand over to Sybe, and both will approximately present for about 30 minutes. And afterwards, we're gonna have a little Q&A session together. And also please feel free to use the Q&A function of Zoom throughout the whole presentation. And we will get then back to it at the very end in the Q&A part. So without further ado, Marius, happy to have you starting the session.
Thank you very much, Fabian. And also thank you, CELEC Germany, for hosting this wonderful event so far. So let me just briefly bring up my slides. So you should be able to see everything now. So I'm happy to talk today about the technological basis of precision fermentation. 30 minutes, it's going to be hard, so I'll just jump right in. So I want to talk about a definition, give a little bit motivation, talk about the benefits of precision fermentation. I want to talk about, of course, the technological basics, just briefly round up everything that is required from biology to technology. Then I want to introduce to you current and future applications, and of course finish with the challenges and also a little bit my perspective on the future potential and the conclusion on that. Before we do this, however, I would like to introduce myself and my group. So in September 22, as Fabian already said, I was appointed the world's first professor for cellular agriculture. We're located at the beautiful campus at the Thun School of Life Sciences in Freising. So Freising Weinstefan, also due to its history in brewery science and dairy science, is one of the worldwide academic hotspots for food research. Now, my current team, and actually still growing team, consists of 2 senior scientists, 2 technical assistants. We now have 7 doctoral candidates, 10 students, give or take, and of course a few international guests as well. Now, when you think about cellular agriculture, many people think this. So animals, or meat without animals. Well, I have to say yes, to some extent. Of course, we do cultivated meat research and everything that comes along. So tissue culture, 3D bioprinting. We also have bioreactor concepts, but we are heavily interested in precision fermentation. And this, of course, is one of the reasons why I am here today. And along with that comes synthetic biology, biosynthetic food protein, and all these beautiful things that you can do. Now, everything is framed by bioprocess engineering, modeling and control, making everything as efficiently or making everything run as efficient as possible, and also develop new bioreactor concepts. Now, when you think about fermentation, you know, traditional fermentation. So most of you, I'm sure, know traditional fermentation. So why do we use that? It has been used for thousands of years for microbial production, preservation, structure and taste improvement of the food that we consume. And it is actually, if you look closer into what people eat, it is roughly 1/3 of our consumed food that is or originates somehow from fermentation processes. Now, in its original terminology, fermentation was used to describe absence of oxygen or processes in the absence of oxygen. Today, we use it for any microbial conversion processes in bioreactors. Now, in traditional food fermentation, spontaneous processes were common, but these are really uncontrolled and not always reproducible. Today, we use mostly defined starter cultures where we know exactly what is happening when we add them to the food product. Now, for precision fermentation, if you want to find a definition, and I wanted to, I did what nowadays many people do actually. I asked ChatGPT, and I asked, hey ChatGPT, how do I explain precision fermentation in a few sentences to a broader audience? And what I got was this. Precision fermentation is a cutting-edge technology that uses microorganisms like yeast to produce specific proteins, enzymes, or other components through fermentation, without the need for traditional farming or extraction. I do not really agree on the part without the need for traditional farming. We'll talk about that later. So it's like brewing beer, but instead of making alcohol, it's used to create a wide range of products, from alternative meats to dairy substitutes, medicines, sustainable materials. This process offers a more efficient and sustainable way to produce important substances, reducing reliance on traditional agriculture and its associated environmental impacts. So it contains a lot of the keywords and a lot of the things I would want to transport when I talk about precision fermentation. I want to elaborate a little bit more, however. So precision fermentation is a rather newly coined term for targeted biomanufacturing processes. In most cases, it's about food, ingredients, additives, excipients, and contrary to traditional fermentation, we use mostly GMOs for that purpose. So it's established for proteins and enzymes and vitamins and organic acids for decades, actually. Now, if you take the first product that fits that definition, it's microbial rennet. So microbial rennet, 1990s, approved in US, 1991 in the EU. This is one of the first examples that really fits that definition. Now, if you look at citric acid that I've depicted here as well, you know that traditionally it has been produced from lemons, but that was actually roughly 200 years ago. Approximately 100 years ago, we found that microbially we have a much more efficient process. And I like this example because it's really very nice example for the entire framework of cellular agriculture. So how more efficient biotechnological processes really have a disruptive potential on existing structures. Now, in the next slide, I want to take you a little bit on a small, don't worry about it, very small journey of the history of industrial biotechnology. So in the traditional industrial biotech, we used mainly glucose, sucrose, starch, or glucose from starch, sugarcane and molasses and so on as substrates. The products that we focused on were complex chiral molecules, pharmaceutical proteins or enzymes, or some economically superior processes. So think back about the citric acid that I just showed you. Now, in 2010, roughly, we had an emergence of the so-called bioeconomy. So bioeconomy tries to reshape the future of industrial biotech so that more lignocellulose-based waste and pretty much non-food substrates are used for bioconversion. It also focuses to some extent on low molecular weight products. Take the 1,4-butanediol that you see. So this is not a natural product. It's not known from biology per se. So very, very nice example for synthetic biology that can be used then in the chemical industry. Now, roughly 8 years later, CELAC and also precision fermentation came up as new technologies. And these also add on to the product portfolio for future industrial biotech by adding biosynthetic and food protein. So this on its own is a huge challenge because for the first time we as industrial biotechnologists are asked to produce food protein in an efficient way, cost efficient, it has to be cheap, and most of all, it also has to be available in huge quantities to have an impact on actual human nutrition. And these quantities are very specific challenges that I'd like to talk about just briefly later on. Now, Of course, it is a challenge, but precision fermentation comes with its own motivation and very, very strong advantages. So for instance, with precision fermentation and what you have seen, food protein, we address most of the benefits that are associated with cellular agriculture. So environmental advantages, health benefits, ethical aspects, just to mention a few. And in theory, it's also possible to utilize renewable resources for that purpose. For microbial protein, we have the general advantage that it's produced fast, we have fast growth, we do have high yields. And the product is also widely applicable. So proteins for food, we know that food, or the proteins in food, are responsible for many of the properties that are very important to us. So it's very widely applicable. And we do have the option of it, the possibility to produce functional and customizable proteins. Think about custom nutrition, specialized nutrition, dietary benefits, health benefits. And one thing that should also be mentioned here, we can increase the acceptance of alternative protein by modifying plant-based products. Now, I would like to begin to introduce to you the technological basics of the precision fermentation. So first of all, we do need a host organism, and over time, biotechnologists have developed quite a portfolio of different microorganisms. So take bacteria, E. coli, it's present in almost all of the microbiologically working labs. We do have Corynebacterium glutamicum. The name tells you already that it has something to do with glutamic acid production, and this is right, of course. For yeast, we do have Saccharomyces, the baker's yeast. We do have Comagataella pfaffii, also known as Pichia pastoris, so a very strong yeast when it comes to protein formation. We do have molds and filamentous fungi and microalgae. Now, how do we actually choose a suitable host if we have all that portfolio, broad portfolio? Of course, first of all, we're checking for natural producers. Are there maybe microorganisms that naturally produce the substance that we need? We check for metabolic capabilities. Does it make sense, or does one of these microorganisms favor production as opposed to others? Then, of course, we look for genetic accessibility and stability and post-translational modifications. This is a huge thing when it comes to pharma proteins, but it's also nothing that we can really neglect when it comes to food proteins. Think about egg proteins. Many of the functions result from the glycosylation. And think about cow milk proteins. Some of them carry very strong phosphorylations. So these post-translational modification is something that we really have to consider when we design the biological background of our process. Now, of course, we need to think about economic viability. Does it scale up well? What are productivity? What are the yields? And we do have regulatory considerations when it comes to what kind of microorganism we use. Now, once we have chosen the host organism, of course, we need genetic information. Let's say one single cow milk protein. And for that, we also have to consider certain aspects here. So for instance, we need to typically translate. E. coli and the cow do not speak the same genetic language, so we need to translate. We need to optimize. We need to optimize codon usage. We need to look at introns and exons and remove the introns that we don't need for our sequence. There are, in many cases, or in some cases, incompatible sequences that we have to get rid of. We need to find one system to introduce it to the microorganism by, let's say, having an expression system, expression vector. Think about promoters, how we drive the expression. Think about regulatory elements, how we can all make this happen. And last but not least, there are regulatory considerations, safety. Where does the material come from? Here we should mention the Nagoya Protocol. So one of the, in 2010, established general frameworks on, well, combating biopiracy. So fair usage of biological material. Now, once we have done this, the microorganism with the genetic information is transferred to a bioreactor. And in the bioreactor, of course, there are different types that we can use. So the most common are actually stirred tank and bubble column reactor. I will talk about this in a second. We need to choose the design depending on whether its intended mode of operation that we want to pursue is actually possible or feasible. So let's say we want to establish fed batch process, we want to have continuous processes. Some of the reactors at certain scales are much better suitable for that. speaking about scale, what is the required size? Depending on the product that we have, what is the effective size that makes sense? Not all the bioreactor types are scaled evenly. Not all the bioreactor types are indefinitely scalable to a size that we want to achieve. And there's, of course, behind all this, there's cost considerations. Now, for the bioreactor types, the stirred tank bioreactor that you can see here is the most common bioreactor. Reason for that is whatever property or whatever demand you have on a bioreactor, if you compare it, the stirred tank reactor is always at least average or better. So mixing mass transfer or the dispersion of gas bubbles, so to say, how it can deal with viscous materials, how it can deal with solids in the liquid. So it's always at least acceptable or better. Now, the other bioreactor that is used is the bubble column bioreactor, and this is really extremely relevant when it comes to large-scale production processes. So you will find stirred tank reactors in almost all sizes in the lab, in all sizes from 1 liter to tens of liters, hundreds and thousands of liters for production. Bubble column, you will typically only find these in very, very large scale. And due to the mode of operation of bubble column, so you put in gas in the bottom, it bubbles up and leaves the bioreactor, and by that it induces mass transfer and it also induces mixing. So there are no mechanical moving parts inside of a bubble column. And this, of course, results in reduced maintenance costs and therefore also in some cases is beneficial to the overall process. Now, if we take a look further, bioreactor is not sufficient on its own. We have some demand on peripherals. So these are, for instance, monitoring systems. Think about additional analytics that we need to detect metabolites, to detect byproducts, for instance, but also to detect the product on its own, actually. Now, peripherals have to align with the process and automation concept. Do we have like an additional downstream process where we need additional vessels, additional piping, additional valves, and so on? Is everything sterilizable? Can we perform the sanitization as required? Can we perform cleaning in place as required? Of course, this will probably be required, but it needs typically additional pumps and vessels to perform that. So something that we have to consider when we build the entire bioreactor production plant here. Now, if we move on, of course, our microorganism would require some nutrients to actually perform the task that we intend. So for the nutrients, what does it mean? We have certain elements, so carbon, nitrogen, potassium, phosphorus, sulfur, magnesium, calcium, and so on and so on. So in terms of sheer amount, carbon sources and nitrogen source are the most important. So for carbon, we commonly use glucose or starch, molasses, so residues of sugar industry, whey, residues of dairy industry. And for nitrogen sources, we use ammonium salts, corn steep liquor, so something that comes from the wet milling of corn after the starch has been removed, and yeast extract, of course. Now, we have to supply the nutrients to maintain growth. We also have to supply nutrients to enable or to fulfill the energy demand that the microorganisms have. We have to consider biological homeostasis. So what does it mean? Always having the correct counter ions for transport and things like that. And of course, we have to account for product formation. Product formation requires carbon source, but if you think about protein as our product, we require nitrogen source just as much here. Now, if we move on, we have certain physiological conditions that we have to maintain. So pH value, temperature, oxygen concentrations, in some cases even more, like salt concentrations, and we observe typically the very distinct optima. So for these optima and also for the addition of nutrients, we need control strategies. Let me just very briefly talk about control strategies. We add substrate to a bioreactor. It's converted to, well, ideally products, of course, but then also biomass, byproducts, and some is lost on maintenance metabolism. So let's say we have pH, temperature, and oxygen under control. We do have implemented some genetic modifications to limit byproducts and to enhance product formation, there is not so much left that we can actually do. So the controlled feeding of nutrients here at this point is the only thing that we can do to affect the product formation. And luckily, product formation is preferred depending on the process and depending on the product at certain growth rates. So by limiting certain nutrients and by controlled feeding, we can actually drive product formation or drive the process into the direction of product formation rather than the other points here that you can see. This is actually one of the research topics that we do in my group, to have a little bit more control over the process, to have these valves that you can see here and introduce them in different spots in the actual metabolism of the microorganism to have more control about that. Now, once our product is produced, process is not finished, we have to, of course, purify. And purification should not be forgotten here, because it usually is known to take a large portion of the costs here. So purification will always depend on the intended application of the purified product and also on the actual product. So we cannot go into much detail here, but just keep in mind that it is a major part of costs here. So let's talk about current and future applications of precision fermentation. So if you think about what products are actually possible, there's quite a lot of them. So it ranges from the milk proteins that you would guess, egg proteins, leather, fur, and silk sweeteners, certain additives to food. So there's a whole range of products that we can produce here. I want to narrow it down a little bit and talk about precision fermentation for alternative protein only here. So now, with all the classes of alt protein here, plant-based, hybrid, cultivated, fermentation-based, we will see now in a second that a product from precision fermentation, specifically microbial protein, is relevant for each and every of these individual categories here. And let's start with plant-based. Now you may ask, what does precision fermentation have to do with plant-based? So a lot of challenges associated with plant-based products. We talked about this 2 sessions ago, actually. So these are low solubility, gelation, cross-linking properties, and so on that don't work the way we want to. And to do this, we can use enzymes that are actually produced by precision fermentation. And in addition, with physicochemical treatment, we can influence the structure, taste, digestibility, so that we can actually, well, have a digested version, digested and cross-linked, altered structure and cross-linked properties to affect all that we later on perceive when we consume that food. Something that is very relevant, and enzymatic cocktails for treating plant-based products have come up as one of the predominant research disciplines in the field of enzymes for food actually nowadays. Now let's move on with the cultivated meat. We discussed this in last week's session. So just extremely briefly, we start from cell material, and then this has to be proliferated. And of course, for proliferation, we do need medium components and or growth factors. So these, you would guess, also originate from precision fermentation and microbial processes. Ideally, this is where we want to go. Part of it can be replaced by plant-based resources, but I'm not talking about this. I'm talking about replaced serum by growth factors, for instance. So this is where we will need at least microbial products to some degree. Then later on, there follows structure and differentiation. And of course, here's the same thing. We will need medium components, nutrients, growth factors for that purpose. One additional application is to have bioscaffold material, which doesn't have to come from precision fermentation, but there are some examples where this also produced a very viable process. And then we end up with the cultivated actual meat tissue that we want, so the structured product. Now let's move on with the hybrid products. Of course, for hybrid products, the prime example for that is the Impossible Burger. So many of you have probably seen it before. It's on the US market since 2016. It's a plant-based burger patty, which intends to imitate meat as much as possible. And it's a hybrid product, soy patty with a protein from precision fermentation. And if you look at the ingredients list, you will find leg hemoglobin, so plant analog to hemoglobin, which is responsible for this slightly metallic, meaty taste that we observe. Now, if we move on to biomass fermentation, we'll see for biomass fermentation, the actual biomass that we produce is the actual protein-based product that we talk about. And there's one approach here that I like to introduce to you. The single-cell protein, as we used to call it, is not a new idea. So in 1968, ICI started producing by methylotrophs, started producing protein by methylotrophs, as a reaction of a United Nations announcement that we will soon be having a protein crisis. So the problem with the methylotrophs that grow on methanol here in that case is that after the oil crisis and in the very late 70s, the entire thing was deemed not sustainable, so they shut it down. But in the meantime, they produced some of the protein product for feed, actually, for animal feed. Now, in 1967, Malo Foods originated from all this, a company Malo Foods, and they kept screening for filamentous fungi that are suitable for human consumption, and they found Fusarium venenatum, something that they market ever since 1985. So as the first meat substitutes from biomass. You will find these in Germany once in a while, not consistently, unfortunately. And let us move on to precision fermentation, so direct and pure production of biosynthetic protein by fermentation. And when we talk about precision fermentation, of course, we will have to talk about Perfect Day. In 2014, the company was founded as Moo-free with a clear intention and directive to produce animal-free whey protein. So 2019, then called Perfect Day, they produced the first commercial product, the ice cream that you can see below here. Very successful, although just a test, a reference product. Now in 2020, they developed their own brands, all held by the Urgent company. You can see on the top right here, Brave Robot ice cream and the Harissa pepper spread. So 2021, Perfect Day products were in over 5,000 supermarkets, and they received a total funding up to today for roughly 840 million euros. So very huge success story so far, at least. So, of course, Perfect Day are not the only companies that are working on that. Just briefly to show you, there are other players in the field as well, and cannot mention all of them. Not all of them are shown here, but I want to mention Formo. So Formo, one of our German companies in precision fermentation, one of the larger companies in that regard, actually. And of course, those vegan cowboys who will have the follow-up presentation here with Sybe, who will present about their work. Still, I mean, biosynthetic milk is a very, very nice example when it comes to the potential of precision fermentation. So first of all, you have the proteins, functional components, taste, nutrition, even added benefits. Think about health issues. So these can be produced in a targeted way by precision fermentation. Now, fat and fatty acids are also extremely relevant when it comes to the taste and the profile that we perceive later on. So these, in the ratio that we need, can actually be produced, you would guess it, by precision fermentation processes. Sugars and sweeteners. It doesn't have to be the RepD, for instance. No steviol glycosides. It does not have to be the RepD, but think about other sugars and sweeteners that are potentially relevant, and many of them can also be produced by precision fermentation processes. And of course, small molecules that further add to stability, to shelf life, add to the taste structure. So all of these make excellent targets for precision fermentation, and also this is why I think biosynthetic milk is an extremely nice example when it comes to how precision fermentation can actually be employed here. Now, let me summarize a little bit. The precision fermentation for food proteins is, first of all, based on a technology that has existed for decades. So it is very flexible and broadly applicable for food. It also has an extremely high potential for future alt protein. And it's heavily targeted by industry and startups. So now we have to answer the question, why is widespread use not established? And I cannot go into all of the details. Sybe from those vegan cowboys will talk a little bit more about the numbers here. But one of the reasons are production hosts are not optimized for that purpose. I've talked about post-translational modifications that are an issue, decreasing production rates, something that we still have to work on in terms of biology. The process infrastructure is usually too expensive. It's not sufficient. There are studies, depending on which work you're looking at, that tell us that only a few % of the overall worldwide protein demand can be met if we take all the precision fermentation capacities in the world. So this is really a huge bottleneck for the future as well. And of course, the cost-efficient downstream processing. So we need technologies for downstream that are cheap enough to justify application for food product. And overall, we need high-efficient process strategies, high-efficient microorganisms together with the process control strategies to achieve what we need and to go in a direction where ultimately efficiency is will result in cost-competitive products for us. Now, if you look at the entire research field, it is extremely interdisciplinary. So from socioeconomic, food law, and so on, to everything that we do is extremely interdisciplinary. And unfortunately, we can already see a lot of skeptics and a lot of, well, negative perception in total of the technology. What we have to consider here, however, is that all available data so far, so try to go out and try to get some data on food product on a large scale. So that doesn't exist in a way that we will need it to make accurate predictions. So the data that we have comes from unoptimized systems. So something that will not allow us any real reasonable or valid prediction for how it will turn out in the future. And food protein, something that you have to consider as well. I mentioned that in one of the first slides. Food protein from precision fermentation is, even though the technology is established, still a very new challenge because we were not used to deal with these quantities at low prices. So this is really something that is a new challenge for industrial biotechnology. And even though there are skeptics, even though it's all unoptimized, it's still a big challenge and it's a new challenge. I am, due to the high benefits that I have shown you, extremely confident, and I'm sure actually that protein from precision fermentation will make an important part, or will make up an important part of the human nutrition in the future. So this is my last statement for today, and I've actually kept in time somewhat. So thank you very much for your attention. If you are interested in a collaboration in any way, joining forces, working together with us, please let me know. And it is my pleasure now to turn over to Sybe from those vegan Cowboys, who will continue today's session. Thank you very much.
Yes, thank you very much, Marius, for this very nice talk. I'm afraid there will be a little bit of overlap, but I will emphasize a few more details. If I'm sharing the right screen now, just a sec. So I will focus on what we are doing here at those vegan Cowboys, and that is the production of casein with the intention to make cheese. And I already saw one question in the Q&A about yield. I will emphasize a little bit about yield and productivity because there are very many enthusiastic papers out there which promise gold. We're also searching for gold, but there are some physical, biological limitations to what you can make from sugar, for example. The first part will be a bit of a repetition, food and fermentation. Maybe I will highlight some different things from what Marius presented. But yeah, that's a traditional introduction of traditional fermentation. These processes evolved basically to avoid food spoilage and to keep it, and to, as time evolved, we started appreciating these fermented products like beer and wine and cheese for their taste, not just only for the nutritional value. Those are, let's say, fermentations of foodstuffs. A second category is what Marius also referred to, the corn, is biomass fermentation. So that is producing microorganisms, mainly with the interest of producing a suitable amino acid, so a nutritious protein composition, which can be used to make food, burgers, or used as feed for animal production. But let's skip the animals for the time being. And then precision fermentation, I put that in brackets because I've been working in fermentation for quite a while, and having read from how it evolved, starting with citric acid, penicillin, glutamic acid. After the Second World War, there was a big hose in antibiotics production. Very large fermentation plants were built. Many of these are no longer used to produce antibiotics because the processes have become so much more efficient that they do not need all that volume. So a lot of these plants are now being used for food production. And same holds true a bit for amino acids, which are produced basically as feed ingredients for animal food. These have also improved so much over the years that also there again you see this wave of fermentation capacity which has become available for new products. And there's an example in Denmark for a really big fermenter which was used to produce lysine, which is now used to produce HMO, so human milk oligosaccharides, an example of precision fermentation, as Marius explained the definition. And for me, let's say the definition is mainly, I guess, that the organisms that are used, in contrast to, for example, glutamic acid, initial penicillin strains, and citric acid, which were all wild type isolates, where you manipulate the fermentation conditions to make them produce the product of interest. In precision fermentation, typically we use genetically modified organisms, where you have introduced a gene of interest, simply said, to produce the product you're interested in. Well, these you've seen, the products of traditional fermentation, which you eat in our daily life. We always start with this to hopefully help acceptance for the more precision type of fermentation products. These are the proteins for searching initially for structure, but also for protein nutritious value. And Marmite is not that nutritious, but it's quite popular in some countries to put on your sandwiches. So soy hemoglobin, I think, is one of the food ingredients after the chymosins, or the enzymes which have been produced over 30 years with precision fermentation for food processing. Soy hemoglobin is an example of an ingredient, and I've actually worked with this the strain producing soy hemoglobin, and it gives you a very nice red fermentation broth. And if you spill it on your lab coat, people think you are working at a butchery. It gives the red colors and the meaty flavor, although I've not tasted the broth, I must admit. Lactoglobulin, perfect day, we will come back to that example. That is produced with a fungus, Trichoderma, while the leg hemoglobin and also the lactoglobulin, which is produced by remilk, is with Pichia pastoris. So Pichia are yeasts, and the Trichoderma is a filamentous fungus, which we'll see later has maybe a slight disadvantage. So these products are on the market, and the next challenge in our perspective is trying to make cheese from microbial produced casein. And this idea was picked up by Jaap Korteweg and Nico Kofferman, who many years ago founded the Vegetarian Butcher, where they actually modified plant proteins to make them useful or mimic meat-like products, sausages, burgers. And they were quite successful, and they sold this company to Unilever, the brand and the recipes. And then they thought, what can we do now with all this money? And then they decided to spend it on a company in Belgium. And they founded the new milk lab, which has later adopted the brand, those vegan Cowboys. And there you can see some of our colleagues, this picture is already 2 years ago. And you might see there our Margaret, so the stainless steel cow, which we will be using to produce casein, we hope. And why would you want to produce animal-free dairy, and more specifically cheese? Well, there are numerous reasons. One is land use. There is a lot of agricultural land used to prepare food and feed, but most is for feed for cattle, either to produce milk or meat. So if you can reduce the reliance on animal milk and meat, you can produce more protein with the same amount of land, or actually give some land back to nature. Specifically for dairy cows, so for cows, is the greenhouse gas impact. Cows produce a lot of methane, which has a 30-fold higher impact on the ozone depletion than CO2. It is gradually broken down, so it's not as lasting as CO2, but the impact is significant. So if you can reduce the number of cows in this world by making casein for cheese in another way, that would be very beneficial for the environment. Likewise, water usage, nitrogen emission, you know, all the discussions on ammonia emissions these days in Europe. And on the other end of the spectrum is animal welfare. I was never aware, but these cows, to be able to produce, to keep on producing milk, they have to calve regularly, which is of course a bit of a challenging situation for a cow to deliver a calf every now and then and get separated from that calf almost instantaneously. Another potential benefit could be that the fermentative protein will be produced without any antibiotics and can therefore be maybe also beneficial for the health impact. Just a few graphs to illustrate where cheese ranks amongst foods. So it is the most water-intensive product per kilogram food product that is known. And in terms of land use, it's just behind several by sheep and beef. And with respect to greenhouse gases, it is even more greenhouse gas producing than, for example, pig meat or poultry meat. So there is really a big environmental impact when you're producing milk, and if you use that milk to make cheese. So that is why Jaap and Nico focused on cheese, as they want to make an impact on our planet. And on top of that, there have been several surveys performed, especially amongst vegetarians and vegans, but also other people, asking if they would consider eating cheese from precision fermentation. And actually, 70% of the people said they would definitely or probably likely buy cheese with fermentative casein. And from my personal surveys, I know that the thing that vegans miss most of the non-vegan products that they do not can eat anymore is cheese. So cheese which has the same taste as real cheese from milk. And the reason why it is so difficult to make cheese from plant proteins is the specific proteins in milk, the caseins. 80% of all the protein in milk are caseins, and these caseins are actually structured. There are different types of molecules in casein micelles, and that's actually why milk is white. Due to these micelles, the light is reflected, and that's why you have the white liquid. And there are, as far as we know, no plant-based alternative protein molecules which have the same similar properties as these caseins. So there the answer is obvious. It is precision fermentation what we would like to go for. This is just a graphic of a micelle, casein micelle, and actually it consists of 4 major different caseins. which are relatively hydrophobic proteins in general, except for kappa casein, which has also glycosylase in groups, so that is a little bit more hydrophilic parts. And all these molecules interact with each other to form these casein superstructures of proteins. Marius already alluded to that. Some of these caseins are phosphorylated, so there's calcium involved in this interaction between these caseins and to form these micelles. And there is a very simple trick used already for centuries in cheese making to disrupt the micelles by a chymosin enzyme. So that's a protease which has a very specific cutting place in the casein molecule, or maybe you should say the casein molecule has a specific place, the kappa casein, where the chymosin cleaves. And as a result, these micelles disintegrate and these hydrophobic casein molecules interact with each other and they form a cheese curd. So that's what is depicted here. So the very simplified version of the cheese production process, starting from milk, and for this milk you need lots of water. But once you have the cow, you harvest the milk, you add some chymosin or rennet that used to be extracted from calf stomachs, but these days it is maybe not in Germany. It can be produced by fungi, or the GMO version, which is actually the same protein as that is extracted from calf stomachs, which is produced with yeast. So that's an example of precision fermentation, production of an enzyme for food application. You add the enzyme, the micelles collapse and the caseins agglomerate. And if you let that sit, you can tap off the water, which has the soluble whey proteins and the other 20% of protein in milk and most of the water. And of course, in milk, there is also fat. So the combination of casein and fat in the curd are then molded and left to ripen, either inoculated with specific cultures, like the Roquefort or the Propionibacterium, to get all the different types of cheeses that you're all familiar with. So starting with 1 liter of milk, which has about 30 grams of protein, you get only 100 grams of cheese, of which 25 grams is casein, 25 grams is fat, and the rest is water and some salts. So that's the traditional process, and the idea of those vegan cowboys was, well, let's replace the cow with a stainless steel cow. And it is, the ambition of our founders that we would actually use grass, and they've done all kinds of calculations that could be possible. But for the time being, we are working with glucose as our readily accessible carbon source from with hedges also has an agricultural source. And the only difference is that we would use the stainless steel cow, so a fermenter, to produce our casein. And then the idea is the cheese making process and the ripening would be very similar to the traditional cheese making process. The technologies that are involved that we have in-house here, molecular biology group, like Marius said, you just need one gene from the cow, but you can do all kinds of optimizations depending on what promoters you use, codon optimization, secretion sequences, signal sequences. So there's a lot of genetic engineering involved to get strains which produce decent amounts of casein, which are then put into fermenters. And another challenge is then isolating the casein in a very cost-effective method to get a curd or a protein. Maybe you can dry it, maybe you don't even need to dry it, which can be used to make either micelles or go directly into cheese. One step back to the hosts, Marius also alluded to them. There are basically 3 categories of microorganisms which are most popular in the precision fermentation field. Bacteria is mainly Escherichia coli, not so much as a production host, but it is used to very quickly make sample amounts of new proteins or modified proteins that you can evaluate in application. Because the strain engineering is so fast, this is the organism of choice for that purpose. But the public perception is perhaps not so readily assumable, so we do not think that you can use E. coli to produce your casein to make cheese. The more obvious suspects, we've seen them, Pichia, a yeast, filamentous fungi like a trichoderma by Perfect Day. They are chosen for different reasons. Typically, the yeast robust fermentation, you can get them in very high cell densities. Pichia is very often still used in startups, at least, with the methanol expression system, the AOX promoter, which is a very efficient promoter to drive protein expression and secretion. But it has maybe its drawbacks that you have to rely on methanol. So there are also other promoters now out there which will function very well in a glucose limited process. So in a fed batch operation using glucose as a carbon source. A big challenge of producing caseins is that they are very sensitive to proteases. You see in the cheese making process that is due to the clipping of casein. Maybe you're familiar with the typical protease assays. These are often based on casein molecules because they are so easily hydrolyzed. They have an open structure and are very susceptible to proteolysis. So in many of the projects in looking to casein production, one of the first steps or part of the development is to remove proteases which are produced by these organisms, and especially the filamentous fungi. They produce quite a few proteases. We have a project together with Leiden University. They have identified, I think, about 69 secreted proteases in Aspergillus niger, and we are evaluating how to cope with those proteases to avoid hydrolysis of the casein. The reason why filamentous fungi are so popular with startup companies and also with funding investors is because of the story about enzyme production with these fungi. And these examples are typically for the production of starch and cellulose degrading enzymes, which are natural products of the filamentous fungi. And yes, they have evolved over a few billion years to efficiently secrete and produce these proteins. And examples in industry, more than 50 gram per liter is typical for these enzyme production. So many entrepreneurs use this number, the more than 50 gram per liter, as their base case to produce casein or any other protein of interest. And I can tell you it does not work like that. It's a lot of work to get into these more than 10 gram per liter production of casein type of molecules with these different organisms. And now a bit on the let's say the fundamentals. Fermentation traditionally was anaerobic, but in this respect we assume it's an aerobic process with a microorganism growing on a carbon source, and it requires oxygen. And that is already a challenge, a solubility. Oxygen solubility is low in water, so you need a lot of energy to create surface area for oxygen transfer from the gas phase to the liquid phase. And Marius already alluded to that, stirred tank reactors or bubble columns. Both can be used with the note that for more viscous broths, probably the bubble column is less efficient. Then I show there the formula. You start with the glucose molecule, you need oxygen, you can produce biomass, CO2. That's an aerobic process. So you're burning part of the glucose to generate energy to produce biomass, to synthesize the biomass and your product. That means you produce heat. So in a fermentation process, the more glucose you can burn or transform into product and biomass, the more heat you produce. So the cooling is really can really be a challenge in an intensive fermentation process. Next to the carbon source, you of course need nitrogen, especially if you want to make protein. There's a lot of nitrogen in protein, to a lesser degree phosphorus, potassium, magnesium, and some trace elements. The nitrogen is often supplied as ammonia, so the base. And that is very interesting because if you consume the base, the pH will drop. And if you use pH control, that way you can automatically supply the required nitrogen for your fermentation, fed batch fermentation. So that's a very typical mode of operation. Glucose as a carbon source, typically a starch hydrolysate, and ammonia as the nitrogen source with the pH control to feed your ammonia. And then, well, you have to, if you want to make a product, you have to have biomass. You have to buy a catalyst to make the protein. And what we always look at is what is now our yield of product on biomass, and the YPX. And this can vary from a very low value to things like lactoferrin to a value of 1. And that is, of course, the case when you're producing biomass as the product. And depending on the product yield, so how much of your glucose goes to product, how much goes to CO2 for maintenance energy, and how much goes to biomass, the biomass yield on glucose will vary, roughly, let's say, between 0.1 and 0.5. 0.5 is when you're only making biomass in a aerobic process, roughly. So then half of your mass of glucose ends up in the form of biomass, the other half ends up in CO2 and water. So if you have these two values, you can define the yield of product on glucose, which is typically the more interesting one, because if you want to make a product, you want to know how much, you need to know how much glucose you have to invest to make that product. And that will determine your price to a large extent of the product. So the theoretical yield of protein, so if you would not need biomass to generate proteins, would be about 0.4. So anybody who's claiming a yield of more than 0.4 gram of protein per gram glucose has probably not done much in fermentation. So that is a nice number to do the sanity check if you see people advertising fantastic yields. So for the dairy proteins, the reported yields until now, the YPS, so the yield of protein on glucose, vary quite a bit from less than 0.002, so 2 grams per kilo, to maybe 0.2, and that depends a bit on the protein. Lactoferrin is on the very low end, and lactoglobulin we've seen quite high yields, and the caseins are until now somewhere in between. So this is about the yield. So how much product do I get out of oxygen? And the second important value is how much what fermentation volume do I need to make that product? Ideally, you want to make a lot of product in a small fermenter. But there comes the limitation of the oxygen, which needs to be transferred. And there is my favorite value is the YOS, so that's the yield of oxygen on substrate. So the amount of oxygen you need to convert your substrate into biomass and product. That value can vary between 10 when you have efficient growth, so then only 10 millimole of oxygen are needed to convert a gram of glucose. So you only produce 10 millimole of CO2, and the rest of the carbon will end up in your biomass and product. But it can also go up to the complete combustion of glucose, and then you will actually burn. You need 33 millimoles of oxygen to convert 1 gram of glucose to CO2 and water. So in a typical fed batch fermentation, you start with high growth rate, biomass production. The YOS starts at about 10. But as the fermentation progresses, the growth rate slows down. There is more and more biomass which can use the same amount of total amount of oxygen and glucose. And this YOS value will increase to something like 25 or even 30 in the fermentation. So it does not make much sense to prolong such a fermentation, because at a certain point you're only burning glucose to CO2. And as an example, in a calculation we'll show on the next slide, we will see that an average value as a rule of thumb is 20 millimole of oxygen per gram of glucose in a, let's say, a process where you're producing a product. And well, to facilitate oxygen transfer, I mentioned the mixing, but also you can apply, and that is typically done in production, a back pressure, so to increase the oxygen solubility and hence the transfer. The typical values of oxygen transfer that you can realize in production fermenters and in lab scale fermenters is 50 to 200 millimole per gram of per liter of per hour, which results in this amount of CO2 with the off gas. And with these values, you can calculate with the YOS and the oxygen transfer capacity, if you take the average value, that you can convert 5 grams of glucose into biomass and product per hour, per liter. So this gives you some numbers with which you can calculate productivity. With a YPS of 0.2, this would mean you can produce 1 gram of product per liter per hour. I see my time is already going fast, so I will go a bit faster. This is an example from the literature, from Perfect Day, from their GRAS notification, which very nicely shows the yield of product on sugar in time. So you see on this fermentation, they don't mention the time, but I assume this is hours. They manage a yield of 0.07, so 70 grams of beta-lactoglobulin from 1 kilogram of glucose, of sugar. This is a fungus, so a low oxygen transfer rate, 40, long fermentation time, 200. If we use the YOS of 20, which was our rule of thumb, we can estimate the total sugar that was fed, 40 grams per liter end of fermentation. And with the yield, we can thereby calculate the titer, 28 grams per liter end of fermentation. So this is not a very mature process, I would say, but they already did an LCA, so trying to show the benefit compared to lactoglobulin from milk. And a very interesting aspect, which Hannah Tuomisto will go in more detail, I think, in 2 weeks' time, is then how you allocate your CO2 production, for example, your greenhouse production to your product. And what better they've done, they showed from our calculations, we can calculate that besides the 28 grams per liter of protein, they also reported a ratio protein to biomass. So we know how much biomass was produced. And they used a peculiar reasoning to compare the impact of the lactoglobulin compared to milk by assuming that all the emissions should be allocated in the fermentation process to both the protein and the biomass, because they said we do not know the value of the biomass, so let's just share it. And in that way, they came to a CO2 equivalent on lactoglobulin of only 2.7 CO2 equivalents per kilogram of protein, while from milk it is typically around 70. If they would have not allocated the CO2 to the biomass, which is actually a waste, unless you can valorize it, this value would be 12.5. But I think Hannah will go in more detail on that in 2 weeks' time. Here I wanted to show a little bit some of these calculations and uncertainties. Land use for milk depends very much on the process. If it's only pasture or if they're in the stable, 4 to 22 square meters is needed to produce a kilogram of milk in the EU. This amounts to, let's say, 10 to 70 kilogram of casein per hectare, while the glucose that can be produced on a hectare is 3 to 4,000 kilograms. Using this low yield from the Perfect Day example, you can already calculate that the land use will be indeed much lower in the fermentative process. And if we would double this yield, so 0.14 instead of 0.7, the land use would be halved again. So this really shows the potential of precision fermentation or fermentation in general to produce protein and to reduce our carbon footprint. So this is how the milk process has developed over time. This is the capabilities we have, those vegan Cowboys. Here is our real stainless steel cow. It's just a standard fermenter, 300 liter, which we'll be using to produce registration batches in the coming year. But we're not the only ones. This is already a slide from a while ago. You see here Formo, started already in 2019. Since then, there have been new colleagues on the field, Bon Vivant, Imaginary. And just recently, so that has also been our motto, how we think the market will be so big potentially, there's no worry about competitors. And we've actually started a collaboration, an R&D collaboration with Formo to speed up our product process and strain process and product development in both companies. And this is some of our accomplishments this past year. This is freeze-dried casein, spray-dried material we produced in a 1,500 liter scale here in Biobase in Ghent. And these are some of the cheeses that we have been able to make. And I think I should stop very quickly.